I'm attending the 17th meeting on acute leukemia in Munich, Germany. And I presented here a talk on BCL2 inhibition in acute myeloid leukemia. So BCL2 is a protein that controls survival of leukemic cells and other tumor cells. And there are several companies right now that came up with the small molecules that can selectively inhibit BCL2 in tumor cells. So one of the molecules we have worked on, it's called ABT199, or commercial name is venetoclax. We have actually developed it from the lab and shown that it has activity in acute myeloid leukemia cells. So we went initially into the phase 2 trial in relapsed refractory AML, but as a single agent it did not have much of activity. And subsequently transitioned to the combination trial with low-dose chemotherapy or hypomethylating agents in elderly AML patients who are unable to get more intensive chemotherapy due to their mainly age. So in that study, which was recently published and presented, the response rate was over 70% at the recommended phase 2 dose. And this combination was recently approved by FDA for the elderly patients. The phase 3 study right now is still accruing patients to show that there is a survival benefit for this combination. In my lab, we're continuing to study this particular molecule and more so the mechanism of resistance to it, what combinations we can use for our future treatment in patients. We have also initiated chemotherapy combination studies in our leukemia department at MD Anderson, and although those are preliminary, they show high activity of this particular therapy. We also use the combination with targeted agents such as FLT3 inhibitors. And overall, we feel that this particular strategy has really changed the landscape of leukemia therapy and will be used in many different combinations across the board to improve the outcomes of acute myeloid leukemia patients.